Long before the pharmaceutical industry existed, before hospitals, prescriptions, and modern medical systems, human beings relied on something far older, far simpler, and far more accessible. They relied on plants, not as a last resort, not out of desperation, but because over time, those plants proved something critical. They worked. Consistently, predictably, and in many cases with a level of precision that modern science is only now beginning to fully understand. Across ancient texts, especially scripture, certain plants appear again and again in healing contexts, not randomly, but with clear, repeated application. Oils used on wounds, leaves applied to skin, resins burned, infused, or pressed into use. For centuries, these references were seen as symbolic or traditional. Today, they are being re-examined under a microscope. And what researchers are finding is striking. Olive oil contains oleocanthal, a compound that interacts with the same biochemical pathway as ibuprofen. Aloe vera, used for skin and wound care, has been validated in controlled clinical trials for accelerating healing and stimulating collagen production. Frankincense contains boswellic acids, now studied in major cancer research centers for their effects on tumor cells. Even the fig poultice used in ancient healing narratives contains enzymes that modern medicine now uses in wound debridement, the removal of dead or infected tissue. This is not coincidence. It is convergence, thousands of years of observation, refined through daily human use, now meeting molecular biology, and aligning at the level of specific compounds, mechanisms, and outcomes. What ancient cultures discovered through experience, modern science is confirming through analysis, and that changes how we understand both. Today, we're examining 12 of the most significant healing plants mentioned in Scripture, each one with a documented historical use, a defined biological mechanism, and in many cases, peer-reviewed research that explains exactly why it works. Before examining the individual plants, it's important to understand the framework that connects ancient healing traditions to modern scientific validation. Because this overlap is not accidental, it follows a pattern. Roughly 25% of modern pharmaceutical drugs are derived directly from plants, or are synthetic versions of plant compounds. This is not a minor influence, it is foundational. Aspirin originated from salicylic acid found in willow bark. Digitalis, used in heart medication, comes from foxglove. Morphine was isolated from the opium poppy. Even advanced cancer drugs like paclitaxel trace back to compounds in tree species. Modern medicine did not replace plant-based healing, it extracted from it. It isolated specific molecules, concentrated them, and standardized their use into controlled dosages. But the origin, the biological intelligence of the plant, remains the same. This leads to a second, more important concept: how researchers decide which plants to study in the first place. The field responsible for this is called ethnobotany, the scientific study of how traditional cultures use plants for medicine. Ethnobotanists don't begin with random selection. They follow patterns of use. When a plant has been used consistently across different regions, across centuries, and for the same condition, it signals something significant. It suggests accumulated empirical evidence, observations repeated and refined over generations. In that sense, traditional knowledge becomes a research map. Scripture represents one of the oldest and most consistently preserved records of this kind of knowledge, not as theory, but as applied practice. And this leads to the third concept, one that modern research is only beginning to fully appreciate: the difference between whole plants and isolated compounds. When a pharmaceutical compound is extracted, it targets a specific mechanism, but in many cases, the whole plant contains a network of compounds, working together. Some enhance absorption, some reduce side effects, others provide secondary pathways of support. This combination is known as a phytochemical matrix. And increasingly, research is showing that this full-spectrum interaction can outperform isolated compounds in certain contexts. The traditional preparations described in ancient texts, oils, poultices, infusions, were not primitive. They were optimized, refined over time through direct human experience with results. Which means when modern science validates these plants, it's not discovering something new, it's confirming something very old. And now, with that framework in place, we can move into the plants themselves, starting with one of the most studied medicinal species on Earth. Now let's move into the next stage. The first plant is one of the most extensively studied medicinal species in the modern scientific literature: aloe vera, a bridge between history and science. Its presence in Scripture spans both symbolic and practical contexts, referenced in Psalms, in burial preparations in the Gospel accounts, and described as a plant of value and cultivation. But beyond its historical placement, what makes aloe significant is the depth of modern validation behind it. This is not a loosely studied herb, it is a clinically examined biological system. The inner gel of the aloe leaf contains a complex matrix of bioactive compounds, most notably ecmanin, along with anthraquinones and enzymes that directly influence tissue repair. In controlled clinical settings, aloe vera has demonstrated measurable effects on wound healing, burn recovery, and skin regeneration. But the mechanism is what matters. When aloe gel is applied to damaged tissue, a semanin activates macrophages, cells responsible for clearing debris and initiating repair. At the same time, it stimulates fibroblast activity, increasing collagen production within the skin. Collagen is not cosmetic, it is structural. It determines the strength, elasticity, and integrity of healing tissue. In parallel, aloe promotes angiogenesis, the formation of new blood vessels, ensuring that oxygen and nutrients reach the site of injury. These three processes, immune activation, structural rebuilding, and vascular support, are the core requirements of effective wound repair, and aloe engages all three. This is why clinical studies across dermatology and surgical research have repeatedly documented faster healing times and improved tissue quality when aloe is used appropriately. Now consider its ancient use. In burial preparation, aloe was combined with other substances not only for fragrance but for preservation. Its antimicrobial properties, now documented, would have slowed microbial breakdown. In earlier texts, its inclusion among cultivated plants suggests it was not rare but intentionally grown and maintained. A plant with consistent application, consistent results, and now a clearly defined biological explanation. Today, aloe remains one of the most accessible medicinal plants available. It grows easily in containers, requires minimal water, and produces a continuous supply of fresh gel, often more active than processed commercial versions, which makes it not just historically significant, but immediately usable. And once you understand how one plant bridges ancient practice and modern mechanism, the next one begins to reveal an entirely different kind of biological power. But that's only the beginning. The next plant appears in Scripture not for its chemistry, but for its contrast. The small humble mustard seed, described as one of the smallest seeds, yet capable of growing into a large dominant plant, it became a teaching tool. A visual metaphor, something small producing disproportionate impact. But that metaphor exists because of a biological reality the original audience already understood. Mustard was not just agricultural, it was medicinal, and modern chemistry now explains why. The seeds of mustard contain a compound called sinigrin. On its own, it is inactive. But when the seed is crushed or chewed, an enzymatic reaction converts sinigrin into allyl isothiocyanate. This compound is responsible for the sharp, pungent sensation associated with mustard. But that sensation is not just flavor, it is activity. Allyl isothiocyanate interacts with inflammatory pathways in the body, specifically inhibiting NF-κB, a signaling pathway that plays a central role in chronic inflammation, cardiovascular disease, and certain forms of cancer. This places mustard in a category of plants that do not merely soothe symptoms. They modulate underlying biological processes. But its most immediate application is more physical, the traditional mustard poultice. Used for centuries across multiple cultures, this preparation involved crushing mustard seeds, mixing them with water or flour, and applying the paste directly to the chest or affected area. The effect was immediate, a warming sensation, increased blood flow, localized stimulation. This is known as a counter-irritant response, where mild surface irritation draws circulation to a specific area, reducing deeper congestion and promoting movement in stagnant tissue. In respiratory conditions, this increased circulation helps loosen mucus, while the volatile compounds released from the mustard are inhaled, acting as natural expectorants. And this is why mustard remained part of formal medical practice well into the early 20th century. A small seed, a fast-growing plant, and a chemical system that activates only when needed. It was functional. The next plant is not introduced in Scripture as medicine, but as a reference point. Coriander, a familiar plant with deep roots in history. In the account of manna, the substance that sustained the Israelites in the wilderness, it is described as being like coriander seed in size, shape, and appearance. This detail is precise. It assumes familiarity, which means coriander was already part of daily life, both as food and as function. Today, that function is being examined through metabolic research, and the findings are increasingly specific. Coriander seeds contain a range of bioactive compounds, including linalool and other terpenoids that directly influence glucose metabolism. In controlled studies, coriander seed extract has demonstrated the ability to reduce blood glucose levels through two primary mechanisms. First, it stimulates insulin secretion from pancreatic beta cells. Second, it improves insulin sensitivity, meaning the body's cells respond more effectively to the insulin that is present. This dual action is significant. Most interventions target one pathway. Coriander appears to influence both, which is why, in experimental models and early human studies, consistent intake has been associated with reduced fasting blood glucose and improved long-term markers like HbA1c. But its metabolic influence is only part of the picture. Coriander, particularly in its leaf form, known as cilantro, has also been studied for its ability to bind heavy metals within the body. This process is called chelation. Certain compounds in the plant interact with metals like mercury, forming bonds that allow the body to mobilize and eliminate them more effectively. While human data in this area is still developing, laboratory and animal studies have shown measurable effects, prompting ongoing research into its detoxification potential. What makes coriander distinct is not a single dominant effect, it is versatility. Digestive support, antimicrobial activity, metabolic regulation, multiple pathways operating simultaneously, and like the previous plants, it remains highly accessible. Coriander grows quickly, prefers cooler temperatures, and produces both leaves and seeds from the same planting cycle. Within weeks, it moves from seed to harvest, making it one of the most efficient functional plants available. A familiar herb, a daily ingredient, and a system of compounds interacting quietly with some of the body's most critical regulatory processes. But the next plant moves away from internal balance and into something more direct, not regulation, but purification. And this is where its historical role becomes impossible to ignore. Hyssop is one of the most explicitly medicinal plants referenced in Scripture, not metaphorically, functionally. It appears in purification rituals and healing contexts, and at critical symbolic moments, from Exodus to the final scenes of the crucifixion narrative. In each case, its role is consistent: to cleanse, to restore, to remove what is considered contaminated. For centuries, that language was interpreted symbolically. Today, it can also be read chemically. Hyssop contains a concentrated set of volatile compounds, most notably pinocamphone and isopinocamphone, along with other terpenes that exhibit strong antimicrobial properties. In laboratory studies, hyssop extracts have demonstrated inhibitory effects against a wide range of pathogens, including Staphylococcus aureus, Escherichia coli, and fungal species like Candida albicans. These are not minor organisms. They are among the most common and resilient causes of infection, which means hyssop is not just active, it is broadly active. And this aligns directly with its traditional use. In purification rituals, hyssop was not consumed, it was applied, bundled together and used to spread liquid across surfaces, doorways, or individuals, functioning as a natural applicator. From a modern perspective, this process would have transferred antimicrobial compounds directly onto high-contact surfaces and into the surrounding air. Not symbolic cleansing, functional sanitation. Even its physical structure supports this role. The plant's dense flowering spikes absorb and release liquid efficiently, making it effective as a natural brush, something that would have been immediately practical in pre-industrial settings. But there is another layer. Hyssop has also been used traditionally for respiratory conditions as an expectorant, helping to clear mucus and support breathing. This aligns with its volatile compounds, which when inhaled, interact with the respiratory tract, loosening congestion and reducing microbial load. So the same plant that was used to purify spaces was also used to clear the body. External and internal application. One function expressed in two directions. Today, hyssop remains a hardy perennial, easy to grow in a wide range of climates, and highly attractive to pollinators. It requires minimal care, yet produces a dense concentration of active compounds throughout its flowering cycle. A plant associated with purification, now understood through antimicrobial chemistry. And as we move forward, the next plant brings that same precision, but applies it directly to one of the most complex challenges in medicine: wound infection, and how to remove it. Now let's move into the next stage. The next plant moves from general antimicrobial action to something far more targeted: the fig tree. In one of the most medically specific accounts in Scripture, a direct treatment is described: a fig poultice applied to an infected boil. The condition is severe, the intervention is simple, and the outcome is recovery. For most of history, that account was accepted without understanding the mechanism. Now that mechanism is clear. Figs contain a proteolytic enzyme known as ficin. Proteolytic means protein digesting. And this matters because infected wounds, especially abscesses, are composed largely of damaged tissue, bacterial structures, and protein-based debris. Ficin breaks those structures down, not selectively in the way a surgical instrument would, but chemically, at the molecular level. This process is known today as enzymatic debridement. In modern medicine, similar enzymes, like papain and bromelain, are used in clinical settings to remove necrotic tissue from wounds without invasive procedures. Ficin operates within that same category. It softens and dissolves the material that prevents healing. At the same time, figs contain compounds with antimicrobial properties, reducing bacterial load while the enzyme clears the environment. Two actions: clearing and control. This combination is critical in wound care. Removing damaged tissue alone is not enough. Reducing infection alone is not enough. Healing requires both. And the fig provides both in a single biological system. Modern studies have confirmed ficin's proteolytic activity and its effectiveness against multiple wound-related pathogens, including strains that show resistance to standard antibiotics. This adds another layer to the original account. What appeared to be a simple plant-based remedy was, in reality, a targeted biochemical intervention, applied without the language to describe it, but with results that made its value clear. Beyond its medicinal role, the fig tree itself is highly productive and adaptable. It grows in a range of climates, produces nutrient-dense fruit, and can be cultivated in both ground and container environments. But its significance here is not agricultural, it is functional. A plant capable of performing one of the most complex processes in medicine, removing what the body cannot heal through on its own, and doing it through enzyme activity embedded in its natural structure. From purification to precision, from general defense to targeted repair. And as we move forward, the next plant expands from localized treatment to systemic influence, affecting inflammation across the entire body through a mechanism that modern pharmacology knows very well. But that recognition came much later. No plant-derived substance appears more frequently in Scripture than olive oil. It is used to anoint the sick, to treat wounds, to prepare the body, and to signify restoration. Its application spans both the physical and the symbolic, but its consistency points to something deeper, a repeated, trusted function. Today, that function can be described at the molecular level. Olive oil, specifically high-quality extra virgin olive oil, contains a compound called oleocanthal. This compound interacts with the body in a way that is both specific and measurable. It inhibits two enzymes, QX1 and QX2. These enzymes are central to the body's inflammatory response. When activated, they produce prostaglandins, molecules that drive pain, swelling, and inflammation. Blocking this pathway reduces those effects. This is the primary mechanism of non-steroidal anti-inflammatory drugs. Ibuprofen works this way, so does naproxen. And aspirin operates within a closely related pathway. What makes olive oil remarkable is not that it reduces inflammation, it's how. Oleocanthal engages the same biochemical pathway as ibuprofen, producing a similar sensory response in the throat and activating the same inhibition process at the enzyme level. Different source, same mechanism. This discovery was not theoretical, it was observational. A researcher noticed that fresh olive oil produced a distinctive throat sensation, identical to that of liquid ibuprofen. That observation led to chemical analysis, and ultimately, confirmation of the shared pathway. But olive oil is not a single compound system. It contains a range of polyphenols, most notably hydroxytyrosol and oleuropein, that contribute additional effects. Antioxidant protection within blood vessels, reduction of oxidative stress, support for cardiovascular health. These compounds do not replace oleocanthal, they support it. This returns to the concept of the phytochemical matrix, the idea that multiple compounds within a plant interact to produce a broader, more stable effect than any isolated molecule alone. The idea that multiple compounds within a plant interact to produce a broader, more stable effect than any isolated molecule alone. In traditional use, olive oil was rarely applied in isolation. It was combined, used as a carrier for other substances, resins, herbs, extracts, enhancing absorption and extending contact with tissue. This is not accidental. Oil improves the delivery of fat-soluble compounds through the skin and into deeper layers, which means olive oil functions not only as a treatment, but as a system, a delivery mechanism, an anti-inflammatory agent, and a stabilizing base for more complex preparations. From enzymatic wound repair, we now move into systemic regulation, affecting pain and inflammation across the body through pathways modern medicine relies on every day. But the next plant goes even further. It doesn't just reduce inflammation, it activates the immune system itself. And this is where the scale begins to shift. The next plant shifts the focus from direct application to environmental influence, cedar. In scripture, cedar is consistently associated with strength, permanence, and purification. It was used in temple construction, in ritual cleansing, and in the treatment of spaces affected by disease. But what appears symbolic is also biological. Cedar releases a group of airborne compounds known as phytoncides. These are volatile organic molecules produced by trees as a defense system, protecting against insects, fungi, and microbial threats. But when humans inhale these compounds, something unexpected happens. The immune system responds. Specifically, phytoncides have been shown to increase the activity of natural killer cells, often referred to as NK cells. These are not general immune cells. They are specialized. Their role is to identify and destroy virus-infected cells and abnormal cells, including those that may develop into tumors. In controlled studies, exposure to forest environments rich in cedar compounds has resulted in measurable increases in NK cell activity, along with elevated levels of intracellular proteins associated with immune defense. And the effect is not temporary, it persists. In some cases, for weeks after the exposure ends. This is not chemical ingestion, it is environmental interaction, the body responding to compounds in the air. Now consider the traditional use. Cedar was burned, handled, and used in enclosed spaces during purification rituals. Combined with other plants, it created an atmosphere, not just symbolic but active. An environment where antimicrobial compounds and immune-stimulating molecules were both present. This is not a single mechanism. It is layered. Cedar contributes immune activation. Other plants like hyssop contribute direct antimicrobial action. Together, they create a dual response system, reduce external pathogens while increasing internal defense. From a modern perspective, this is a coordinated intervention. From a traditional perspective, it was simply what worked. Cedar's role is not to treat a specific wound or condition directly. It is to shift the body's readiness, to increase its capacity to respond, and it does so without direct contact, through inhalation, through presence, through environment. Today, cedar and its related species are widely available and adaptable across climates. Even exposure through essential oils or wood environments can release measurable phytoncide levels. A tree associated with purification, now understood as an immune modulator. And as we continue, the next plant returns to direct treatment. But with a compound so influential, it eventually became one of the most widely used drugs in human history. The connection is exact, and the implications are difficult to ignore. The next plant is not just medicinal, it is historical. The balm of Gilead appears in Scripture as a symbol of healing so well established that its absence becomes a question. If the remedy exists, why is healing not taking place? This was not poetic exaggeration. It reflected a substance widely known for its effectiveness. What was not known at the time was the chemistry behind it. Today, that chemistry is understood. The balm of Gilead, particularly in its accessible botanical equivalents like balsam poplar, contains a compound called salicin. Once ingested, salicin is converted by the body into salicylic acid. This is the same foundational compound that led to the development of aspirin. Not similar. Directly related. Aspirin, acetylsalicylic acid, is a modified version of this naturally occurring molecule, designed for stability and controlled dosing. But the biological activity originates from the same source. Pain relief, inflammation reduction, fever control. These are not modern discoveries. They are modern refinements of something observed thousands of years ago. Traditional preparations of balm of Gilead, often made from resinous buds, were used topically and internally for joint pain, muscle inflammation, and wound care. The resin also contains antimicrobial compounds, adding another layer of protection in damaged tissue. So the plant does not act through a single pathway. It combines analgesic and antimicrobial effects, relief and repair. And this explains its value in ancient trade systems. It was not just a local remedy, it was transported, exchanged, and preserved across regions because of its consistent results. But there is another important distinction. In its natural form, salicin is delivered within a broader phytochemical matrix. This can influence how the compound is absorbed and tolerated by the body, often producing a slower, more sustained effect compared to isolated pharmaceutical versions. This difference matters. It highlights the distinction between extraction and experience, between isolating a compound and using the plant as a complete system. Today, the connection between balm of Gilead and aspirin is one of the clearest examples of how traditional plant use directly informed modern drug development. A line that can be traced without interruption, from resin to compound to pharmaceutical standard. And as we move forward, the next plant represents something even more complex. Resins studied not just for inflammation or pain, but for their potential role in one of the most serious diseases in modern medicine. And this is where the research becomes significantly more advanced. Frankincense is one of the most extraordinary plants mentioned in Scripture. Gifted to the infant Jesus, it was valued not only for its rarity, but for its therapeutic potency. In the first century Mediterranean world, frankincense was considered more precious than most metals. Carried across deserts, traded along perilous routes, and reserved for sacred rituals, it was medicine, perfume, and symbol all at once. Modern science has now verified why it was so highly prized. The key compounds are boswellic acids, particularly AKBA, acetyl-11-keto-beta-boswellic acid. These compounds have demonstrated potent anti-inflammatory and anti-tumor activity. Studies at institutions such as MD Anderson Cancer Center and Johns Hopkins University have documented frankincense extract selectively inducing apoptosis, or programmed cell death, in cancer cells without damaging adjacent healthy tissue. The mechanism is precise. Boswellic acids inhibit 5-lipoxygenase, or 5-LOX, an enzyme that drives a particularly destructive inflammatory pathway associated with arthritis, cancer, and inflammatory bowel disease. This differs from conventional NSAIDs like ibuprofen, which target COX enzymes but have minimal effect on 5-LOX. Frankincense thus complements standard therapies rather than duplicating them, explaining its persistent presence in traditional medicine. Beyond cancer research, boswellic acids have been studied for their ability to reduce joint inflammation, support immune health, and even mitigate anxiety. These effects align closely with the ceremonial use of frankincense in sacred spaces. Burning resin releasing volatile compounds into the air, producing a calming, protective environment. From a botanical perspective, Boswellia sacra grows in arid zones. For modern cultivation, container growth allows enthusiasts to maintain a living specimen even outside its native environment. But its medicinal relevance extends far beyond the plant itself. The convergence of ancient ritual, empirical healing, and modern pharmacology illustrates a recurring pattern. Plants selected for spiritual and ceremonial significance were often chosen precisely because they conferred real, observable health benefits. Frankincense serves as a bridge between worlds, ritual and science, sacred and empirical. It demonstrates that the ancients were not guessing. They were observing, experimenting, and recording results over centuries. And yet, even as frankincense's properties are recognized today, the story continues. Its resin is still being tested in new clinical contexts, including neuroinflammation, autoimmune disease, and complementary cancer therapies. But that's only the beginning. Myrrh, often paired with frankincense in Scripture, holds a unique place in both sacred and medical history. From the Nativity story, where it was a gift for the newborn Jesus, to the crucifixion, where myrrh-mixed wine was offered. To alleviate suffering, this resin has been consistently associated with preservation, healing, and pain relief. Its chemical composition is remarkable. The key compound for anudesma 1,3-DN interacts with opioid receptors to provide analgesia. Unlike pharmaceutical opioids, it offers pain relief without the risk of respiratory depression or addiction. Beyond analgesia, myrrh is a potent antimicrobial agent. It acts against bacteria, viruses, and fungi, making it invaluable in wound protection and preservation. This property explains its widespread historical use for embalming. Modern science confirms these effects. Clinical research demonstrates that myrrh tinctures effectively treat periodontal disease, reducing inflammation, inhibiting pathogenic bacteria, and promoting tissue regeneration. These findings reflect a direct continuity between ancient practice and contemporary clinical application. The resin contains additional terpenoids and sesquiterpenes that enhance its antimicrobial and anti-inflammatory profile. These compounds are volatile, meaning inhalation during traditional burning rituals could deliver systemic benefits, supporting respiratory health and providing a calming protective atmosphere. Myrrh's enduring relevance is also evident in modern integrative medicine. It is still employed in dental clinics across multiple countries and explored in wound healing research, particularly where conventional treatments struggle with resistant pathogens. Cultivation of myrrh for contemporary use requires warm, arid conditions. Outdoor growth is suitable in zones 10 to 12, while containers allow broader access in non-native regions. Even a single plant can yield small but potent quantities of resin for educational, experimental, or therapeutic purposes. The convergence here is profound. A plant selected in ancient times for ritual and symbolic reasons turns out to contain compounds with scientifically validated pharmacological effects. This illustrates a principle repeated across many scriptural plants. Empirical observation over centuries identified bioactive compounds long before modern science could explain them. Myrrh stands as a living testament to the intersection of tradition, observation, and scientific validation. It shows that the ancients' understanding of healing was not mystical guesswork, but the result of careful, sustained empirical insight. And this is where timing becomes critical. Mandrake, or Mandragora officinarum, is one of Scripture's most intriguing and symbolically rich plants. Its roots, often anthropomorphic in shape, drew attention in ancient texts as objects of fertility, healing, and spiritual significance. In Genesis 30:14-16, mandrakes appear as a valuable bargaining tool between Leah and Rachel, tied directly to fertility and family continuation. Song of Solomon 7-13 highlights their aromatic qualities, reflecting both sensory appeal and medicinal value. Scientific exploration confirms that mandrake's efficacy was more than symbolic. The root contains tropane alkaloids, primarily scopolamine and hyoscyamine, compounds with documented pharmacological effects. These alkaloids exert sedative, analgesic, and antispasmodic actions, supporting traditional use for pain relief and calming effects, and even fertility-related applications in historical herbal medicine. The plant's pharmacology is complex. Scopolamine affects the central nervous system by modulating muscarinic acetylcholine receptors, producing mild sedation, antispasmodic effects, and anti-nausea properties. Hyoscyamine works synergistically to reduce smooth muscle contractions and relieve pain. These mechanisms align with centuries of empirical observations recorded in herbals, emphasizing the root's value across diverse cultures. Mandrake's dual role as both food and medicine is notable. It demonstrates that ancient societies were acutely aware of dosage and preparation. Raw roots could be toxic, whereas tinctures, infusions, or cooked extracts provided safe, targeted benefits. This careful management reflects sophisticated traditional pharmacology. Cultivation today requires Mediterranean-like conditions, well-draining soil, full sun, and protection from frost. Zones 6 to 9 are ideal for outdoor growth, while containers extend accessibility to colder climates. Even a small plant produces roots with concentrated alkaloid content suitable for educational or historical botanical purposes. The mandrake story also exemplifies a broader theme in scriptural plants. Careful observation over centuries led to the identification of effective remedies. Modern phytochemistry validates these ancient insights, confirming that many biblical references are not mere allegory, but empirical documentation of bioactive compounds. From the fertile valleys of the ancient Near East to contemporary botanical gardens, mandrake illustrates the enduring intersection of culture, scripture, and pharmacology. The plant embodies the ancients' deep connection to natural cycles, reproductive health, and the empirical wisdom embedded in scripture. And now, the stage is set to explore the final plant on this journey, one whose fragrance, medicinal value, and spiritual symbolism have resonated across millennia. Pomegranate, known as the fruit of life in ancient texts, is a symbolically and medicinally significant plant in scripture, symbolizing fertility and healing. References appear throughout the Hebrew Bible, where pomegranates adorn temple garments and sacred spaces as signs of abundance. Modern science confirms its potential, with seeds rich in antioxidants and polyphenols that support cardiovascular health and reduce inflammation. Studies show regular consumption can lower blood pressure and improve heart function, while also offering antimicrobial properties. Growing them is simple. They thrive in full sun and well-drained soil, even tolerating drought, making them perfect for your home garden. Incorporating this ancient fruit connects us to a lineage of healing. This completes our 12-plant exploration, bridging ancient wisdom with modern wellness.